covering for this presentation is cellular motility driven by assembly and disassembly of actin filaments. It's written by Thomas Pollard and Gary Borzi. They performed this research at Yale University in New Haven, Connecticut, and in Northwestern University of Chicago, Illinois. So as for the contents of this article, one of the most clear points that they make in it is the abundance of actin filaments and general actin proteins in eukaryotic cells, most particularly in muscle cells, with those being some of the most abundant proteins. And from there they go into the two types of actin. So there's G-actin and F-actin. G-actin being the monomer form of it, which is globular and just kind of generally more simple. F-actin is more stringy, and it's composed of a bunch of subunits of G-actin formed along a kind of rope-like structure called a filament. To be more exact on that ropes structure, it's a double helical. And so to get more precise into how G-actin and F-actin relate, so G-actin concentration affects the rate of growth of F-actin, and one of the ways in which it does this is through what's called the critical concentration of G-actin, which is whenever there's so much G-actin, it almost instantly starts to form F-actin until it reaches back to that initial concentration that it was at, which is quite peculiar and rather unique to actin. As well, in G-actin, it's formed by two domains that are separated by a cleft that contains a nucleotide, which is usually either ATP or ADP, and this is largely relevant to how G-actin forms into F-actin. ATP forms much faster than ADP. And the last point is in relation to barbed and pointed ends that relates into the structure of F-actin. So pointed ends is where the cleft of the G-actin subunit faces away from any of the other actin subunits of the filament, whereas the barbed end is where it faces towards another one of the subunits. So the barbed ends are much better for the growth of the F-actin, as you could then just add another actin subunit onto that because it's already facing towards an actin subunit.